As you can see, we are in the exhibit center here. This is a place where a lot of industries are showing their support for the human proteome organization and the proteomics community. This is a very intricate relationship of academia and industries here. Where a lot of the instrument vendors and the chemical reagent vendors, everybody comes together and try to share their most recent innovation in the field of proteomics. So let's have the Rohan Thakur here with us for an interaction on the HUPO industry perspective of proteomics.
Proteomics has existed for about, let's say, 20 years or more, where a lot of fundamental work has occurred in the past in understanding fundamentals. And I think in the next few years, you're looking at the application of proteomics in what's known as translational work moving towards the clinic. So you have to apply what you learned with a few samples to doing large cohort studies. So I think the application of proteomics and what we have learned over the past 20 odd years and moving it to larger sample cohorts to understand how disease works either in a diagnostic or a prognostic manner is going to be the future, where we connect applications of proteomics into the clinic, in the clinical research environment. And these studies are already ongoing. And at HUPO this year, you saw Rudy Ebersol present this, very nicely, where the impact of running sample studies of more than 100 patient cohorts is starting to have an impact in proteomics. So that would mean taking proteomics from the laboratory and moving it towards basically into a research clinic, into a hospital. Right, that's an excellent question, because everything has to relate to biology, right? Or the disease that happens in the human body and not in artificial conditions in the laboratory. So single cells, for example, they're going to change once they're outside of the body or in the laboratory. So how do you capture that in a chemical state? So if you look at stuff like native mass spectrometry, where you are doing the analysis of large intact native proteins to see how these structures behave in the gas phase versus studying them in the liquid phase, like with NMR or some technique like that. It's a big leap to see how these proteins in their native conformations, either as complexes, exist and how they would interact in the human body. So it's a huge step. Instrumentation is, I would say, evolving in that direction, and it's an evolving field. Difficult to do, but the first steps have been taken. That's great, right? So if you look at the differences between diagnosis and prognosis, right? Prognosis is how will the disease progress in the future. Diagnosis, what's wrong with you now. So identifying biomarkers has been easy to do. Validating the biomarkers has been almost impossible to do. But as you start doing larger cohort studies, this is going to change. And I think the big difference between mass spectrometry in proteomics in the last 2 decades versus proteomics in the future is going to be the ability to study large patient populations all over the world, right? So infectious diseases, large populations, India, Africa, Asia, connected to studies that you're seeing in Europe and America. So I think large patient cohort studies, exactly what happened in the genomics world, where you could do large global genomic studies, is going to be the answer to look at predictive diseases, diagnosis, prognosis using proteomics and mass spectrometry. In what proteomics in mass spectrometry requires is harmonization of standards. Basically, we need standards in proteomics that do not exist right now, but it's reflective of the field being in its infancy. Right? It's almost like microscopy in the 1800s. So as the field gets more mature, you're going to start seeing harmonization of standards, standardization of procedures so that the results can be compared. Right? Search engines are being developed in everyone's individual laboratories. So bioinformatics is going to play a big role, and you can see the rise of machine learning in bioinformatics, in proteomics. And if you look at proteomics, it is a clear definition of big data. So machine learning is going to play a big role in hopefully harmonizing the results that come from standardizing how you run the samples, how you prep the samples, and present them to mass spectrometry. My advice would be read up on the fundamentals. You must understand how the tool works. So if you get a firm understanding on how mass spectrometry as a technique works, then you can use it to apply in biology and the treatment of diseases, right? So understand the tool and then apply it to medicine or biology or whatever you are trying to solve. Right? Mass spectrometry is a tool, and how you use it depends on how well you understand it, and then using that in that manner. So fundamentals would be my advice to the young scientist or the young researcher.